to list 10 functions of blood. You'll see that these functions could be combined. You wouldn't necessarily end up with 10, but 10's a nice round number, so I like 10. And of course, most of these functions, or at least half of them anyway, have to do with transport. Because the whole idea is that we have the cardiovascular system that pumps the blood through the body so that we can transport things. So transport of oxygen, transport of CO2, transport of nutrients, transport of waste, hormone transport, right? All of these things done by blood because it's able to move. We can also give a function for blood in that it's important in acid-base balance. We'll talk more about that a little bit later. It's involved in temperature regulation. You knew that, right? So that we can change the flow of blood to different parts of the body. Water balance is another function, as is the production of antibodies. And finally, coagulation. Right? So there's 10 functions of blood for us. If we take a look at blood, we can put it in a test tube, and the cells or formed elements will settle out, or we can put it in a centrifuge and centrifuge that. Those of you that have lab got to do that on Monday. And when we do that, we find that the formed elements go to the bottom and the plasma ends up on top. The plasma will have kind of a straw color to it. And it's mostly going to be water. On average, it makes up about 55% of a person's blood. As I said, mostly water. There's nutrients, there's waste. We find electrolytes in there. And we also find some proteins in there. One of the proteins happens to be a protein called Fibrinogen, which is an important clotting factor. If we remove Fibrinogen from plasma, we then call the plasma serum. Right? So we give it a different name because serum won't clot and plasma can. In addition to that protein, Fibrinogen, we find a group of proteins that we call the albumins. And the albumins have a number of functions. They are important because they create osmotic pressure. Also, they serve as carriers in the blood. And then we have a group of proteins that we call the globulins. And we're interested primarily in a group of them called the Gamma-globulins. And your Gamma-globulins are your antibodies. And those will become important to us when we get to the immune system. All right, so the fluid part, plasma. The rest of it, then, the rest of the blood, we call the formed elements. When we look at the formed elements, well, plasma makes up about 55%. The formed elements are going to make up about 45%. When we look at the formed elements, we're going to find that there are erythrocytes, leukocytes, and thrombocytes. Erythro means red. So these are your red blood cells, the erythrocytes. Leuko means white. So these are your white blood cells. And the leukocytes can be subdivided into those that have granules, so-called granulocytes, and those that don't have granules, what we call the agranulocytes. And then finally, what we call the platelets or thrombocytes. Thrombo means to clot, right? So the clotting cells, although they're not really cells. Here's a diagram. We'll get this more in a few minutes, but it's trying to show there's your red cells, here's some granulocytes, and here's the agranulocytes and the thrombocytes. We're going to come back to each group individually. So let's look at the erythrocytes first, the red blood cells. When we take a look at these red blood cells, you know from anatomy that the major function is the transport of oxygen. You may not know that they're important in transporting carbon dioxide as well, right? So transport oxygen, but also transport carbon dioxide. When we centrifuge our blood and we separate the plasma from the formed elements, almost all of those formed elements really consist of the red blood cells. And so the percentage that the red cells make of the total blood, we call your hematocrit. Okay? The percentage of the red cells that make up all of your blood, we call that the hematocrit. And those of you in lab got a chance to measure this the other day. And it turns out if we look at hematocrit values for males and females, they vary slightly. So that for a female, we expect hematocrits to be about 42% plus or minus 5%, and for males, about 45% plus or minus 5%. All right, so this is going to be the amount of red cells in your blood. If you look at a red blood cell under a microscope, the red blood cells have no nucleus. Right? They are anucleate. Right? So here's a nice diagram trying to show this. No nucleus in the center. They are in the shape of a biconcave disc. I like this artist's drawing. I thought that was pretty nice, right? So some artists are pretty talented to end up drawing a red cell like that. So a biconcave disc with no nucleus, and they are packed full of hemoglobin. By not having a nucleus, there's more room to have hemoglobin in there. And by having the biconcave disc shape, we get a lot of surface area without taking up much space in the capillaries. All right, so large surface area, but not using up a whole lot of space. They're packed full of hemoglobin, I mentioned already. I told you on Monday that there's a protein in our muscle cells that we call myoglobin that appeared red, but it had one iron molecule. When we look at hemoglobin, it turns out there are 4 different chains, and each chain has an iron molecule. So there are 4 different iron molecules in a molecule of hemoglobin. The hemoglobin molecule can join with oxygen, and so that's why we want our red cells full of oxygen or full of hemoglobin, is so that we can have this very simple chemical reaction. We can have hemoglobin plus oxygen going to oxyhemoglobin. Okay? Hemoglobin plus oxygen goes to oxyhemoglobin. Of course, that occurs in your lungs. When you get back in the tissues, it runs backwards. And so the oxygen will be released from the hemoglobin. When hemoglobin combines with oxygen, it takes on a nice bright red color. Okay? A nice, really bright red color, about the color of David's cap. Right? So this really nice kind of bright orange-red color when it's full of oxygen. If you ever get a chance to see oxygenated blood, you won't forget it. Right? It's really nice bright red. When it gives up its oxygen, it turns kind of a dark maroon color, more like Jordan's sweatshirt. It's nice that they sat together today, right? It's the color that you're used to seeing when you stick your finger, right, and blood comes out. So that's oxygen or deoxygenated blood. So when the hemoglobin has given up its oxygen. At no time is the blood ever blue. And so I'm sorry that if your grade school teacher told you that blood in the veins is blue, it's not, right? The combination of the maroon color with your veins makes that nice blue color. I can guarantee you that if a phlebotomist stuck a needle in a person's vein and sucked out blue blood, they would get very excited, right? It never turns blue. All right, so life cycle of a red cell, about 4 months, plus or minus. So 127 days, right? But about 4 months. The making of red blood cells has a special name. We call that erythropoesis. Poesis means to make. So the making of red blood cells, right? Erythropoesis. It occurs in your red marrow. And it might surprise you to learn that the red marrow that you have is relatively limited in terms of where it's found in your bones. Your axial skeleton certainly has red marrow, right? So the skull, the vertebral column, sternum, ribs, your pelvis will have red marrow. But your long bones aren't going to have red marrow unless you're under the age of 25. So some of you might still have red marrow in the proximal ends of your femur and your humerus, but that's it. Right? After about age 25, those areas don't have red marrow anymore. So we're really talking about the axial skeleton and the pelvis, right, for having the red marrow where erythropoesis can occur. Erythropoesis occurs at a tremendous rate, 2 to 5 million cells per second. Right? Now, that's not a misprint, right? 2 to 5 million cells per second. Think about that. Let's go low end. 2 million cells a second. Every minute, 120 million red cells. How many minutes have we been in class already today? Right? I mean, it's huge, the production of the red cells. Right? Very, very rapid in terms of cranking these cells out. In order to make red blood cells, you need some important nutrients. They're pretty obvious. You're going to need amino acids because you have to make hemoglobin, right? The globin part is protein. We need the iron. Maybe not so obvious, but you need folic acid and vitamin B12. And it turns out folic acid and B12 are involved in DNA synthesis. And so in order to make DNA, you've got to have B12 and folic acid. And of course, we're making red cells at a tremendous rate. When they're first made, they have a nucleus. We'll look at that in just a second. So you need B12 and folic acid. There's other trace minerals that you need, things like copper and cobalt and some other strange things, but those are the major ones. So the way that we make these red blood cells is that in our red marrow, we have a stem cell that we call, a number of them, not one, right? We call these stem cells Hemocytoblast. Right? Hemocytoblast. Do you remember in anatomy what that term blast meant? To make, right? To make. It makes whatever's in front of it. Right? So hemocytoblast means blood cell making. Right? So these stem cells are hemocytoblasts. They're found in our red marrow. And we're not going to go through all the different steps, but it's trying to show you that here's the hemocytoblast, certainly has a nucleus, and here's the lineage for making red blood cells. There's the end product. So we start out with a nucleus in the red cell, but as the red cell matures, eventually the nucleus is lost. I do want to know one individual step here, so I'm only going to mention one, and you'll find out why in a second. So the second to last step before we have a mature erythrocyte is something called a reticulocyte. The second to last step is called a reticulocyte. And I mentioned there's the word. The reticulocyte is important. It shows the breakdown of endoplasmic reticulum. And I mention it because reticulocytes actually end up being in the circulation, actually being in blood, but they only last out there in the blood for about a day. However, if we start to see lots of reticulocytes showing up in the blood, it indicates that the person is making blood cells at a very high rate. And so it can indicate that there's something going on, right? That they've had anemias or a hemorrhaging or something. So we'll learn just that in the intermediate step, the reticulocyte, along with, of course, the hemocytoblast and the erythrocyte. So that's the life cycle, about 127 days. We're making it 2 to 5 million per second. 2 to 5 million, that's a pretty big range. So let's think about what kind of control mechanism must exist to change that. So let's say that you go this afternoon and you donate blood. This morning you were making red cells at 2 million per second, but if you just went and donated blood, you better not be making it still at 2 million per second this afternoon, right? You've just lost a unit of blood, so you'd better be able to increase the rate of production. And it turns out that the control mechanism for how fast erythropoiesis occurs is governed by a hormone called, cleverly, I think, erythropoetin. Okay? So the hormone that governs this is erythropoetin, and it is produced by hypoxic kidneys. Okay? So hypoxic kidneys will produce erythropoetin. hypoxic is a clever combination of hypo and oxygen. So when your kidneys have low amounts of oxygen, they will be stimulated to release erythropoetin, which will increase the production of red cells, trying to counter the fact that you don't have enough oxygen in your blood. Right? So a nice little feedback loop here. Right? So the kidneys sense the lack of oxygen, release erythropoetin, increases the production of erythropoiesis, increase red cell, lowering or correcting the oxygen deficit.
Negative feedback loop?
Another negative feedback loop. Gosh, it's been a good 30 years at least that this particular hormone, erythropoetin, the gene for that was isolated. Genentech was able to do that. And once erythropoetin was available to help people with severe anemias, right, because we could give them erythropoetin to help them produce the red cells, but once that was available, it didn't take long for athletes to say, gee, if I took erythropoetin, I'd have higher hematocrit values, and I would be able to do more exercise. Right? And so it has been a drug that's been abused by athletes. Whole professional bicycling teams have been kicked out of the Tour de France for using erythropoetin. In the Australian Olympics, quite a ways back, but in the Australian Olympics, the Chinese women runners came out of nowhere, long distance runners, and started winning races. When they showed up in Australia, they had suitcases full of erythropoetin. And so they were all promptly sent home, right? Because they were using erythropoetin to increase their red cell counts. The earliest use of erythropoetin by athletes that really is documented was in the early 80s. They had a couple of professional bicyclists from one of the Scandinavian countries. I don't remember if it was Sweden or Norway, one of those countries. Two young men in their early 20s, both died of heart attacks. And they said, gosh, what's going on here, right? I mean, early 20s, you're a professional bicyclist and you're dying of a heart attack? That doesn't make sense. Well, what had happened is they had used erythropoetin so much that their blood got so thick that it caused the blood to clot, of course, in the heart. And these athletes then died of heart attacks from excessive use of erythropoetin. Okay. Destruction of red blood cells. Maybe before I leave that, would it surprise you to learn that endurance athletes oftentimes will train in plastic tents that have low amounts of oxygen in them? Another trick that people do is they cover their beds at night with plastic tents, and then again have low amounts of oxygen in their bed while they're sleeping. Make sense? Right? So they can increase their erythropoetin levels naturally so that they won't be accused of illegally using erythropoetin. It's the same as training at altitude, right? Essentially the same as training at altitude when they do this. So I said the red cells live in your body about 127 days. If they have no nucleus, they're not going to be able to control structure and function, right? So the cells are going to not be able to repair. They wear out, and when their membranes start to wear out and are broken down, you have specialized phagocytes in your liver and in your spleen. So these are fixed phagocytes, macrophages, that don't travel around. They just sit there in your liver and your spleen. And when these red blood cells show up and they have an abnormal shape because their membranes are wearing out, the macrophages will grab those red blood cells and break them down, right? Destroying them. Now, I said that the red cells are being produced at 2 to 5 million per second. How fast do you think you're breaking them down? 2 to 5 million per second, right? They better be about equal, right? Now, if you lost blood, then you're going to have to be higher on the production side, but, right? So, huge rate of breakdown, 2 to 5 million per second. Yes?
If you donate blood like a stated amount, approximately how long would it take to replenish that?
Yeah, so it's just about 6 weeks to get it completely back up to where it's supposed to be. The fluid actually you'll replace within about 3 hours, but the red cells about 6 weeks. All right, so you're breaking them down at this tremendous rate. You're making them at a tremendous rate. Hopefully it would make sense to you that the body does a very good job of recycling the nutrients, right? So that you can reuse most of the substances required for making red blood cells. So we're going to break that hemoglobin down into the heme and the globin. The globin can be chopped up and reused as amino acids, right? So we'll reuse those amino acids. We're going to recycle the iron, right? So make new the heme part of the hemoglobin. But you do end up with a waste product, and the waste product is called bilirubin. I went to 2nd grade with Billy. He's a pretty nice kid. Right? You guys know Billy? Bilirubin? Don't know him? No? Bilirubin is a yellow-green colored waste product. When you get a bruise on your arm and the red blood cells leak out into the tissues, macrophages come and clean that up, and when they leave, you're left with this kind of yellow-green color. The liver is constantly pulling bilirubin out of the blood and depositing it into the bile system. And so it makes your bile that greenish yellow color that you're used to seeing when you see the bile, right, and the gallbladder or the bile ducts. When the bilirubin is secreted from the liver and the gallbladder into the small intestine, it eventually ends up in the colon, and you have bacteria in your colon that work on the bilirubin, changing it from a green-yellow color into a brown color. Have you ever wondered why your feces are brown? Huh? Here's an experiment you can do on your own at home, right? So right, usually I say, don't do this on your own home. But this one you can do on your own home. Pick a day, maybe between now and next Monday, and eat only white foods for the whole day, right? So white rice, white potatoes, white cheese, white milk, everything white for a day. When you get up the next day and you take a look at your poop, guess what color it's going to be? It's still going to be brown. Okay? It doesn't matter that you ate white foods all day long. Your poop's still going to be brown because the bilirubin is turned into this brown color by the bacteria in your colon. If any of you have ever had small children, right? You've had babies. When the babies first start pooping, it doesn't come out brown because they don't have any bacteria in their colon. It comes out kind of a greenish yellow color, right? Kind of a shock when you first see that. But eventually bacteria begin to accumulate, and so you get the brown color. So, when you go home today and they say, what did you learn in school today? I say, I learned why my feces are brown. Okay. Food coloring certainly changes it. I remember when Nathan was a little kid, he was over here at the Child Care Center, and on St. Patrick's Day, back in those days, they would put green food coloring in the cookies and the milk, everything, right? And so Nathan came home, I changed his diaper. Jeez, right? I mean, it was all green. But all right. Anemias.